I'm Miguel Alcalde. I'm research professor at the CSIC, the National Research Council of Spain. And I'm also founder of EvoEnzyme, which is a spin-off company stemming from my lab. And I like to consider myself as a biologist by training, but a protein engineer by devotion. What inspired me the most in enzymology, well, when I was making my PhD many years ago, I was in a European project trying to engineer some enzymes, and the process was kind of frustrating because we were using rudimentary techniques. But things started to change when I had the great opportunity to make my postdoctoral stay with Frances Arnold at Caltech. She's the inventor of directed evolution, and she was awarded with the Nobel Prize in Chemistry for this invention and for the development she achieved. And the point is that during those years at Caltech, her lab was at the boiling point of ideas and innovation. Well, possibly the lab has been all the time at this point. And that experience in her lab changed my life, gave me focus. Possibly that's why I'm right now making protein engineering. Well, biotechnology is, I would say, a broad concept. Actually, you can see biotechnology surrounding us in every single aspect of our lives. In my case, I consider myself very lucky because I've been involved in many different projects designing enzymes for applications ranging from bioremediation, the removal of pollutants, to biopulp, pulp biobleaching, organic synthesis, or biomedical devices. Currently, we are pretty much involved in engineering enzymes for the pharma and chemical sectors. Well, directed evolution is a revolutionary strategy to tailor enzymes, in general terms proteins, metabolic pathways, or even whole microorganisms with improved traits. Basically mimics the Darwinian algorithm of natural selection, but at the test tube. So you introduce random mutations, recombination events, and you couple this with screen or selection methods in such a manner that you design customized biocatalysts. And I would say that directed evolution has influenced a lot protein engineering in general terms. If you look back 20 years ago and now, most of the experiments in terms of enzyme engineering are being currently done by directed evolution. I would say that automation and in general terms instrumentation are vital for directed evolution experiments, because at the end you have to screen large mutant libraries. So definitely all these advances are helping directed evolution to go further. It's true as well that apart from instrumentation and automation and all this kind of equipments, it's needed mutant library construction, and right now also we have the push of artificial intelligence so that we can reduce the screen effort. I think that biotechnology has changed a lot. If we take a look back 20 years ago and see how biotechnology is developing, it's just fascinating, right? I'm very bad in predictions, but I would say that taking into account the problems that we are facing in terms of sustainability, circular economy, to reduce the carbon footprint. Especially in Europe, for example, we have this 2050 Green Deal. We are supposed to get climate neutrality by that date. So we have to change our habits, and biotechnology has many things to do in this field. So we have to change our way of producing chemicals, our way of producing energy, how we manage the waste. All these things must change, and biotechnology is a strong strategy to work with. Well, I would say that directed evolution, I've already said in my lecture, is influencing not only protein engineering, but broader disciplines like synthetic biology, metabolic engineering, systems biology, and actually it's affecting by directed evolution we can harness all the power of natural evolution, but at the test tube, to create different products which are covering different areas like blue, green, white, red biotechnologies. And of course, the future of directed evolution, I think, is going to be dominated by the discoveries and the findings in artificial intelligence. So I hope that in the near future, perhaps in 10 years or so, we could just make directed evolution assisted by computers, and we just need to screen a few tens of variants at the end of the process. So everything is going to be faster. Well, leaving enzymes aside, because we've engineered many enzymes, possibly the proudest achievement is to see my students growing up and see how they get positions either in the academia or in the industry. And also the foundation of our spin-off company, EvoEnzyme, which is great that is working so well. Well, yes, right now we are working really hard with peroxygenases. We are going to try to achieve chemical reactions that are not described in textbooks. Also, we are involved in plastic degradation and upcycling, designing enzymes for this big issue in Europe and in general terms all around the world. And in other projects, like we are trying to design a smart wound dressing for monitoring in real-time infections with enzymes. Yeah, thank you.